from Harvard University, MIT. He completed a postdoc in the Orthopedic Research Laboratories at the University of Michigan at Ann Arbor. He originally joined Columbia as a mechanical engineering professor in 1996, and then later joined the Department of Biomedical Engineering. He has been promoted through professors, named endowed chairs, and he is now currently the department chair at Columbia. He also runs the Bone Bioengineering Laboratory, focusing his research interest on the micromechanics of bone, computational biomechanics, and mechanobiology of bone. He has won countless honors at all stages of his career, Young Investigator Awards, Established Investigator Awards. He's won funding from the NIH and the NSF, as well as several other societies and foundations. He has been named a fellow of AIMBE, of the American Society of Mechanical Engineers, of BMES, and the International Academy of Medical and Biological Engineering. He was a founder and served as a co-editor-in-chief of Cellular and Molecular Bioengineering, CMBE, which is an international journal for BMES and one of the journals of the special interest groups for people who do cellular and molecular bioengineering work. He has served on many review panels for NIH, NSF, NASA, and his research has also been supported by the Whitaker Foundation. He has been the chair and leader and awardee of many different boards and many different societies. And he has just a really wonderful career and is a fantastic person. I've gotten very lucky and got to meet him at some conferences a few years ago. Yeah. So now I will turn it over to Dr. Guo, who is going to present on bone, bioengineering, microstructure mechanics, mechanobiology, and beyond. Thank you, Dr. Guo.
Thanks so much for kind invitation and also kind introduction. And is very glad to meet you guys on Zoom. But ever since I become chair about over 3 years ago, and then I visited UAB very shortly, a kind invitation by John. And that time I made it, it's John's cardiovascular fantastic program. And then John also invited me on the external advisory board. And that one I totally missed, stuck into airport somewhere. And then, but I was happy to arrive, able to join the nice dinner John hosted. So he's very glad to give a presentation. And then we hang together for this tough year. And then it's coming. I think, I'm getting, I just vaccinated on Tuesday, got my second dose. I think, this is also going to come in soon. So I'm going to talk about some of my research. So I'm studying bones, as you can say, bone bioengineering lab, so don't do anything else. And hopefully you can see, it's actually keeping me busy enough. So bone is actually quite ancient, right? So ever since you have animals and you have bones, all the animals actually come from fresh water, from fish. So when the fish made a decision many million, hundreds of million years ago to decide to go to land, and then there was a tremendous evolution. I think we'll still try to understand. So one of the evolution, of course, you don't want to be killed. So therefore, you want to become light. So the one process is really losing bones. So therefore, even humans has been evolved. We're still pretty early in terms of the process. We still try to get as much as possible, get rid of bones. That's really governing the bone biology and mechanics here. So therefore, the mechanical forces, you can see my former student, Rachel, was a former professional dancer. And, skeleton, of course, is very important. Without skeleton, she's not able to do that. And also the dancing itself, mechanical stimuli is also very important making the one. So I'm going to talk about mechanics, microstructure, focus on microstructure, mechanics, and at the end also look at why the structure is like this, because there are mechanobiology behind it. So this is the bone I'm interested in. So this is a cross section of vertebral fractures, not vertebral fracture, not fracture yet, vertebral. So this is very normal. This is a very normal vertebral. You can see they have very nice, seems disorganized structures in there. But if you're looking very carefully, just by eye, you can see they were kind of sheet-like structure, and then also like a kind of rod-like structure. So therefore, these structures is going to be, I'm going to talk about, very important. And then another thing you can also point out is it's not totally disorganized, right? You can see along the axial direction, a lot of bone actually have preferred orientation on that. So during the aging or during the post-menopausal period, and then you lost a lot of bones. When you lost in bones, you can see that the bottom one is still normal, but lost a lot of bones, right? The most striking one you can see here is loss in bones. But if we're looking very carefully, they have transitions. Those kind of sheet-like structure, plate-like structure is almost gone. This one you can see mostly is by raw-like structures. So that will result in one reason, resulting a lot of osteoporosis-related fractures. One of them is called vertebral fractures. So if you look at the X-ray on the right, and they can see the top and bottom are normal vertebrae, okay? And in the middle one, you can see is squished. So that's actually a sign of vertebral fractures. So most of them actually you don't know about it. So you can see this is a 3-generation ladies from Hong Kong, and then you can see the signs of the change in the back. Actually, it's a sign actually have vertebral fracture. Of course, not just changing doesn't look nice, but actually can causing troubles. Can can compresses the nerve and have pain as well as paralysis. And then there's also another disease which is also related to, so osteoporosis actually have a lot of drugs now and they're able to, to treat in the osteoporosis patient. So one thing also is on the other side is osteoarthritis, the joint disease. So you originally thought it's cartilage disease. And then in this process, the bone itself looks very opposite, right? Because you can see on the right, the bottom, that's actually a osteoarthritis bone underneath the cartilage, and then you can see the tons of bones there. On the left side, of course, the picture is showing in the loss in bones in osteoarthritis. And then there are more medical biology understanding right now, and a lot of changes in the bone actually starts with resorption. And then so how you lost in bone early and relate to very lot of bones later, I think that's actually also very interesting. The very interesting questions we have. And then in addition, the bone is actually alive. So this is a very, very old hypothesis is bone mass and microstructure follows mechanical loading. And then it is truly is there, right? So if you look at this professional tennis player, his playing arm not only is bulkier with muscle, and then the diameter of the radius and then is actually about 20 to 30% bigger and thicker. So therefore, the mechanical use is definitely, well creating one. And then I should update this one, pick new picture with from the Mars, and then also probably put a picture of, Bernie Sanders sitting in there. So that's what I'm going to do, update my picture. So we're going to send in people to Mars, and then the Mars. But on the Mars, you have gravity, but the problem is we travel until to figure out in the science fiction movie, have this big rotation centrifuge, and then you lost in 2% bones per month. So you're taking about 12 months to fly to Mars, and then take another 12 months to fly back, right? And 6 months going there and 6 months going back. So therefore, you're going to lose him close to 24% bone loss. And then that you can see the consequence of loss in that much bone is actually very detrimental to the ones here. So therefore, not only mechanical stimuli is important, and it's actually have consequences as well. And then so in the future, hopefully we're going to go to Mars. So I'm very interested in this microstructure bone. Fortunately, there are techniques able to providing non-invasively the microstructure here. So one of them is called microcomputer tomography. It's just a CT, but just have a high resolution. So you can go into 5 microns and 10 microns of resolution. And this same machine and using humans is called a high resolution peripheral quantity computer tomography. So essentially just a micro CT we use in humans. We can only measure in the distal end of the limb. So therefore we call PQCT. And then the resolution probably is about 100 microns, even though the voxel size is smaller, about 60 microns. So from that, as I mentioned, I'm very intrigued about this microstructure, and I come from mechanics background. So therefore, I know these features of plate-like structure, we call them trabecular plates, and the rod-like structure, call them trabecular rods, are very important. As also I mentioned, orientation is also critical in terms of how to quantify this. So take us many years, actually, we developed this very innovative three-dimensional segmentation technique is called individual trabecular segmentation, we call it ITS. So showing here is just the process, and through the three-dimensional pattern recognition skeletonization, we're actually going to be automatically segment this individual trabecula out. So you can see one of the rod-like structure has been segmented automatically in this yellow in the bottom pictures in there, right? So if we're able to do that, then you can describe how much bones in there, percentage bones there. That's called a bone volume fraction, essentially percentage bone divided by total volume. So therefore now we can quantify how many plate bone volume fraction that they have in there. It's called a PBVTV, and they can also calculate how much rod bone volume fraction there. It's called rod BVTV. And then you can also quantify the orientations of each individual trabeculi, longitudinal, oblique, and transverse, right? And then not only that, you can actually calculating axial, we call it axial bone volume fraction in terms of how much bone volume is actually aligned in particular orientation. Turns out the bone is not really disorganized. Plate and rod structures actually are made such that most plates are in the longitudinal directions showing on the left, and the most rod are in the transverse direction distributed showing on the right, on the plate on the left and the rod on the right, right? And you can also quantify individual trabecular thickness and also the connectivities, how much, the density as well, how many number of plates, total number of plates divided volume, and how many rods you have in here. So this is a really basic of a biomechanics of trabecular bone, or the spongy bones we're talking about. Plotted in here, Young's modulus describe how stiff these materials are. And they can see the yellow one is the bone volume fraction, product about volume fraction. You can see how much bones in there explain quite a lot, how much the mechanical property is. You can see, R squared is 0.94, 94% explained by the changes in bone volume fraction. Of that, if you're taking on its plate, is PBVTV, plate bone volume fraction in green, you can see equally describe this variation as well. And then not only that, if I calculate both the plates and rod, but in that particular orientation in three directions, okay, in the vertical axial directions, we call it axial bone volume fraction. You can see almost explaining 100%, 0.99, of the electron module. So therefore, the take-home message is plate is really supporting mechanical loading. Plate bone volume is very important, and also the orientation, how much in the axial direction is also very important. Right? So this is a three-dimensional image analysis technique. Then early on, people asked me a lot of questions, how accurate they are, right? Because you can see on the top is the macro CT images, and the bottom and the middle is actually plate and rod representations, right? So then the question of how accurate they are. So now we want to answer that question biomechanics. Okay, so therefore we can do computation as well as mechanical testing. Is in the middle is based on our plate rod assumption with created models, and then the bottom is called a voxel model, means the micro CT images of the original image, and they will convert 100% to a model, and then we can assign them same tissue properties for both models. So then we can compute and also compare with experiment. You can see here. In the green is experiments, and in the yellow so-called voxel model. That means there's no assumption about plates and rod. And then the blue are plate rod finite model. You can see the finite model of plate rod predicting exactly same slope, which is how stiff the materials are. And also when you see the curve, green curve beginning to bend it, I think you can see the plate and rod curves actually falls better. So it means the predicting strength a little bit better. Right? So we do not play any tricks in here. Both models are using same exactly elastic property. All the properties are the same. So therefore, we can comfortably say now, the trabecular bone is made of a plate and rod based on the biomechanical perspective, right? So it's all in there. And then, as I said, making it more interesting is now you actually can apply to human studies. This is called, HRPQCT. I mentioned is essentially a micro CT. And then the first generation have a 82 micron voxel size, and the second generation right now have 61 micro voxel size. But itself actually can do 45 micron scanning, but in general, they do not want us to do that. And they can scan your wrist, your ankle, and also the new version actually can also scan knee as long as you're not too tall, not too big, right? So you can see that's a picture of me, and then I scan my legs and quite a few times. So this is representing images of what it look like. So you can see a scout view, you can see the middle is normal bone look like, you can see the fine, this porous trabecular bone structure inside. Definitely, you can see, you can identify in the fracture group that actually have deteriorations in microstructures in there. So now, not only you can quantify the microstructure, of course, you can also do, we call the virtual mechanical compression. You can determine the whole bone stiffness, as well as you taking the spongy bone out, the trabecular bone out, you can also calculate the stiffness as well. So you can do computations on this object. So we have done many studies working with our endocrinology colleagues to try to see whether they can predicting fracture better, right? So this is one example in the vertebral fracture, and then we have some patients with vertebral fracture without the vertebral fracture, matching them together. And normally in clinic, you're using a called DEXA to measure the bone mineral density or area bone mineral density. So these patients, between the fracture and the control, we know they have spine fracture, but the bone density measurements in the spine, which is indicated by LS, number spine, statistically there's no differences, right? So therefore, the bone density measurements is actually not a very good predictors of who is going to have fracture, who is not going to have fracture. And then the predicted percentage essentially just a little bit over 50%. So, you know, it's a 60%. So the 60% time that you can predict in fractures. And you can see if we measure, they are using HFPQCD, quantify the radius and tibia, and then do the ITS analysis, and also we can look at changes in the cortex as well. You can see in the control one, you see more greens than the one in the fracture group, right? So green means place, and definitely you can see in these measurements, you can see there is a transition into the fracture group. There is transition from plate-like structure to rod-like structure. Indeed, if you quantify them and then compare to the fracture group, and they can see there's significant loss of plate bone volume fracture, loss by plates, and then maybe some changes in the rod bone volume fracture, but not major, only in the radius. But you do have significant loss in the plate rod ratio, indicating there is a transition from plate-like structure to rod-like structure. And interestingly, you know, the axial bone volume fracture, I said, you know, it's really supporting mechanical loading. And unfortunately, you also have a significant 20% loss of axial bone volume fracture in there. And then, you know, that loss is mostly by PTBN. So what it is loss by plate numbers. So therefore, you actually not just by thickness, actually those number means the number of trabecular plates are gone, right? And then there's also disruption of the trabecular bone connections. So plate to rod, plate junction, plate to rod junction, and rod junction also has been changed, right? And then this consequence, there are significant changes in the trabecular bone stiffness as well as whole bone stiffness. So in theory, we could use this technique to predicting fractures better. But of course, you know, the study is ongoing and it's more challenging because the problem is you rarely have fracture in your ankle, and you do have fracture in the radius, but most of the fractures in osteoporosis is in the hip and spine. So therefore, you know, radius is not exactly hip, you know, it's not the exact spine and the tibia. So that's really, you know, the accuracy is not really, we cannot measure it, but we cannot measure the side. So if one of you, new generation, able to develop a high resolution techniques, able to measure microstructure in the spine or in the hip, then I'm 100%, you know, convinced, you know, we actually can make prediction fracture pretty reliably. So in summarize, you can see in the osteoporosis in fracture, you're lost in trabecular plates, you're lost in axial aligned trabeculi, you have plate to rod conversion, you're also lost in the trabecular connectivities. Looking at the other end, as I mentioned, there is an osteoarthritis. The challenge of osteoarthritis, we don't know how osteoarthritis occurs. The risk factors, for example, if you have ACL tear, you're actually going to have a fracture. You're going to have osteoarthritis in 10 to 15 years, unfortunately. That's what happened now. And then, but we actually take this disease is not uniform, right? This is on the picture showing here is doing the total knee replacement. You can see on the medium side, which you can see a lot of red, essentially those are the ones that don't have any cartilage at all. But on the lateral side, on the left, actually the cartilage looks normal, right? And that's in general what it is. The medial side is more severe. So therefore, even with this severe osteoarthritis, they have to do total knee replacement. And then, but we can take advantage, do cross-section studies. And then when we're first taking a subchondral bone, which means underneath the cartilage, we found out that actually different from for example, in the femur, right? So as I mentioned before, in the femur, the longitudinal plates are in the longitudinal direction, which is the right bottom, and the rod mostly in the transverse direction is right top. And then if you look at those numbers, right, majority of those one are plates, right? 25,000 are plates. And then on the top is 6,000. So therefore, you have more plates and then supporting load. And then, so one thing different in subchondral bone is, first of all, the rod distribution is the same. So most of the rod are in the transverse direction. But the plates, I will call, you have two peaks. One is in the axial direction. Another one is roughly in the transverse direction, or close to transverse direction. So it's like a camel has two humps. And then another one is also interesting, is different now. If you look at the number, the rod number is 1,000, and then the plate number is 400. So therefore, subchondral bone actually is reverse. It's actually have more rods than the plates. And on the other hand, it makes sense, right? Because it transition from very soft cartilage to a bone. So therefore, that kind of supporting more compliant structure is very important. So as we take advantage of this total joint replacement, and then you can take in samples, as I mentioned before, in the medium side is very severe. In the lateral side, actually is pretty mild. So therefore, we can take in the samples, CT images from different places. So this is a cross-section of micro CT scan. So even with a micro CT scan, you actually can also get in contrast for soft tissue as well. You can see the shadows of a normal cartilage in there. And then you look at the bottom, it is osteoarthritis. In the medium side, it's very severe, right? You can see there is intense of the trabecular bone underneath the cartilage, and also the cartilage is almost gone in the medium side. On the right side, it's not normal, okay? It's absolutely not normal. So because you can see there's actually signs of osteophytes, close to here. And also you can see there is a fracture in the subchondral plate. But if you just look at the density, you probably did not see too much density in there. Another thing also important to find out is actually the thickness of the cartilage actually looks normal, right? Actually, even though they're not the thickness wise, actually normal. And indeed, if we quantify by histology, we find out the several regions in the medium side, actually, they actually are normal. So you can show these yellow thing here, you quantify the cartilage thickness, that's actually normal, right? So by score. So therefore, they are normal. They're no different from the other controls as well. So we taking samples from these regions, we're going to pay attention to these three regions here. So that's why I intentionally block out the other one. Other one also have OA already. So we call these places are intact because they don't have osteoarthritis yet. So if you look at the bone one fraction, you do not see any difference as a quantified. And then if you look at plate bone one fraction, they're not different either. And then if you look at the mechanical property of trabecular bone, they're also not different. So that's really people have debating, whether osteoarthritis is a cartilage disease or is a bone disease. Because anytime you see the cartilage change, there's a bone change, right? Anytime you do not see cartilage change, there's no bone changes here. But I will say there's something more fine you have to measure. That is by rod bone one fraction. You can see those areas, even the bone density is the same, but the rod bone one fraction has been significantly decreased, and the plate to rod ratio is significantly increased, right? So this is the opposite of what happens in osteoporosis, right? Osteoporosis, you have plate loss, and then you have plate rod ratio decreasing. And this change is not by plate number, it's actually by loss in rod number. You can see the rod number has been significantly reduced, right? in here. So that's what we found something interesting. And then why you actually do not see any change in the thickness, in the bone density? Because you lost in some rod number, but the remaining rod and plates begin thicker. So you can see the thickness begin to increase. That's why the total bone volume fraction is not significantly increased. It's not significantly decreased. And on the medium side, as I said, everywhere is osteoarthritis. We look at all the regions in here, right? So therefore now you see the significantly increased bone volume fraction, and then now the plate bone volume fraction is significantly increased. If you look at the mechanical property, they're very, very stiff. They're actually very, very stiff. They're several times increased, about commonly a little more. But interestingly, you can see the deficit in the rod bone volume fraction still there. Okay, they still have a deficit in rod bone volume fraction, and then the plate to rod ratio is going to the roof, can be increasing 200% or 300%. It's really changing, more plate-like structure. And then interestingly, also the rod number density still deficit. So therefore, this is not normal. This also is not a longitudinal study, just cross-section study. But it do suggest something interesting could be happen is there's something happens, rod loss in the mild form of OA actually could happen first, right? And then, the density of the plate, of course, increase. So what do we want? We don't have a human studies yet. We're trying to do that. But there has some evidence suggesting the bone changes may preceding a osteoarthritis, right, from our analysis. First of all, we can take in the guinea pig. So guinea pig, one of the guinea pig you buy most time in the pet store, as well as the guinea pig, you order from the animal research, is this guinea pig, hardy, hardy guinea pig, Dunking hardy guinea pig. They actually will develop osteoarthritis, spontaneous osteoarthritis, in the age of 4 months. So second month, 3 months, you can see the cartilage actually is normal. But interestingly, we'll be observing in the second month, 3 months, you can see there is a significant loss of rod numbers. Okay, the beginning loss in rod already. So suggesting some changes early before they actually have a cartilage change. So therefore, our hypothesis is really the rod loss could be early sign of changes of condylar bone. And one of the hypotheses, as say, in normal conditions, you have a mixture of plates and rod, you have actually a lot of rods. Somehow you're losing the rods by abnormal bone biology. When the rods are gone and they cannot replace them, therefore, the only thing can do is really gradually making the rod thicker, and especially the plate thicker, right? And then when the plate is thick to enough, then actually changing the forces in the cartilage above them, and eventually getting the vicious cycles. You lost your cartilage. When you lost your cartilage, you become more stiff in bone, so on and so forth, right? So that's our hypothesis. We want to do proving that. And then, as I said, the new generation, if you're not too tall, I'm not too tall, and then you actually can also scan your knee as well. Of course, it is an awkward position because it's not designed to scan your knee. So therefore, you have one leg, have no place to play. So therefore, I have to put my legs up in there, and so therefore, scanning is not very comfortable. But we get really beautiful images in here. You can see this is my scan, so therefore, can show to you without any violation of IRB. And then I have to say, I really admire my bone microstructure. Looks beautiful. When you say that, and of course, you joke on side, you can actually do analysis taking the femoral side and also taking tibia side. So our hypothesis is applying to now is let's look at an ACL tear, right? ACL tear, we know that actually loss in bones. We know in ACL they lost in bones, but then later on in 10 to 15 years, they actually develop OA, right? So our hypothesis is that maybe in the ACL injury, you actually lost in rod. When you lost in rod, it's kind of you changing this picture permanently. Indeed, we have done some follow-up studies, and then first scan, visiting one, is immediately after ACL tear as soon as possible. Second one, in general, is 1 month later, just before the surgery. And then the third one is 1 month after surgery because they come for the checkup. You can see the first one, bone volume fraction, is not new. They lost in bones. And then the plate volume fraction on the bottom left does not change, right? And then you see there is significantly increased plate rod ratio by losing the rod bone volume fraction on the right top. So therefore, there are changes consistent with our hypothesis. So we're very excited that you're really extending these studies, looking at the possibility in a way. So taking everything together, so the trabeculae bone, which is very important, you want here, is just a bunch of trabeculae plates. Plates and rods, add them together, equal trabeculae bone. And the trabeculae plates, axial trabecula, are very important in supporting load in the vertebrae, in the hip, but also if it's too much, it may not be good in the subconjugal bone, because if you not want subconjugal bone, have a lot of plates in there. So this ITS technique actually is a very good morphological biomechanical one here. And then in osteoporosis, you have plate loss, plate to rod conversion, reduced connectivity. And then osteoarthritis, you have loss of rod, thickening plates, reduced connectivity. So too little, too much, either of them are bad. And so we have a chance of collaborating with our collaborators, and then he introduced John Benizicki, a doctor at Columbia Endocrinology Department, and introduced me studies. Is we know different race actually have different risk of fractures, right? So one of them is a hip. Chinese in general is relative to Caucasians is about 30% of the risk. If it's a white, Caucasians are 100% of risk. And then interestingly, Japanese and Koreans, we're very close to each other, actually have 70 and 50%. So that's actually pretty interesting. So what we found out now is we did first in a premenopausal woman, which is before the menopause, and then we're looking at the bones, right? So using the HRPQ-CT. And then we just applying them to ITS segmentations, and then turned out the Caucasian white woman and Chinese woman really have a complete different bones. You can see here the Caucasian white have a lot of red, which is rod, and Chinese actually have a lot of green or plates, right? And then if we quantify them, there's no difference in the rod, but the number of plates in the Chinese are double the amount of one in the white. And then also the axial bone one fraction is also almost double. So therefore, it's really the difference is in the plate bone one fraction. Chinese woman has a lot more plate-like structure. And then this plate-like structure is because they have more plate numbers. The number of plates is more in Chinese women. That one translating into a biomechanical advantage. So the Chinese woman have stronger bones than the Caucasian woman. So we got this one very interesting. And then now we're putting all the different samples into here, different age group. Turns out it's actually a very sensitive differentiator of Chinese versus Caucasians. So we do not look at the pictures, look at genetic stuff, right? We're just taking measures of plate rod ratio. So if the plate rod ratio is less than 0.62, we classify correctly 80% of the white, and plate rod ratio greater than 1.34, we classify 81% of Chinese. And then the ROC curve AOC is 0.78. And then if we have a medical device which have a AOC greater than 0.7, actually you can apply clinically. So therefore, in theory, you can just use this plate and rod ratio, classify who is a Chinese Caucasian. So therefore, we have fascinating interest do these studies in China because different region and different ethics. And then also I introduced the US, but we're never able to get it, areas funded. So, but it also got me interested in genetics and also anthropology, how the evolution is, right? Because this is related to, I was talking about Chinese, Korean, and Japanese are very close to each other. My hypothesis is maybe the bone actually is different, right? Because they're so close, because we all come from Africa, and then we settle in the south of China. And early on, about 10,000 years ago, and the yellow one is migration of Japanese, okay? And then about 6,000 years ago, 5,000 years ago, the blue is migration of Koreans, right? So you can see Koreans come from actually northeast part of China, and then that's where they came the ones here. And then you can see if you look at how the distribution, they're all on the top. That's where Koreans, Japanese, all in there. And then so how the differently have risk of have the fracture, I think they were fascinating. But I have not studied that one yet. But if you look at the red arrow pointing another group, it's very, very close to Caucasian, but also from China. So those are the minority Uyghurs. There are 70% of Caucasian, 30% of Asians. So those you can see, they're very, very Caucasian looking in the green area, which is called the New Territory, Xinjiang. So we are very interested to do the study. But I spent like 5 years to try to convince people to do this study. I say, you just need to spend $600,000 by this HR PQC, then we can get a study done. But nobody's buying that machine for me. And then I say, you're building a multidisciplinary genetic medicine institute, different disciplines working together. I'm interested in bone, osteoarthritis. You can be interested in cardiovascular. You can be interested in cancer, right? Sounds great, but nobody's want to do this one for me. But finally, I changed the one. I need to deliver this scanner to the location we want to do. So I designed this trailer, and this trailer has a CT machines in there, and also have a living quarter. So it's in Chinese because it's in China, but they essentially have beds in there and also have kitchens, have bathrooms in there. So it's a super trailer. So that's the trailer we designed, and then you can see, I cannot drive it there. I wish I could drive. And then you can see there's an HRP scans in there. So everything's ready. And then unfortunately, the COVID hit. So we are delayed for 1 year now. I have graduate students ready to study. So I'm looking forward till the COVID is gone and then can go back to these studies here. So this is turns very interesting, right? Not only, related genetics, race, and health, but actually it's interesting. We can also using these bones and do the ITS analysis and the segment of different, not just mammals, just segment of primates. You can look at, we look at the bones differently, right? So Sam is my graduate student, just pretending he's a halibut hanging, in the bar, right? And they can turn. You can see we actually can use this technique to segregate in the gorillas, chimpanzee and pan, and the pongo, which is red gorilla, and then Homo sapiens, of course, us, and the halibuts, right? Using this kind of microstructure, actually we can separate the different primates, separate together. That's quite interesting because in the anthropology, they actually use a very low-tech thing to do, right? That you see the ox in there, that's essentially the shape of the jaw, right? Look at how teeth was looking like, and then they're able to segment different people so here. So we have the data analysis in the year, but we don't know how to write that paper. It has been 6 years. We need to publish that paper. Of course, I have the question people ask me, why you have plates? Why you have rods, right? So we believe plates is by mechanical loading, but we never had any data to supporting that. So I'm going back to this one here is mechanobiology, how the bone responds to mechanical loading. Of course, it goes through cell, goes through tissue, goes through the structure, and then eventually go to whole bone. So that's very hierarchy ones here. Then you need to do cell studies, in vitro expand studies, and in vivo studies in here. So before we talk about the results, I want to introduce you to how bone is formed. That's the two processes forming bones. One is called remodeling, means formation follow resorption on same space, and is regular for the bone maintenance and usually balanced, but it can lose in both. And another one is called modeling, which actually can have uncoupled formation. We call them anabolic, and also uncoupled catabolic. Okay, so why this one is important? I'm not going to get into detail because they have different bone biology behind it. Different cell actually is working. Okay, so in the bone remodeling, you actually need a coupled osteoclast versus bone cells. But both process, modeling and remodeling, can gain bone and also loss in bones. Okay, so both processes, either remodeling or modeling, depends on here. And then, so the question we have for now, when you subject to mechanical loading, where and how much bone is formed, resolved in response to mechanical, that's first question. Okay, and then where the form, right? Is it by plates or by rods? Right? That's one way we're reading that question. Second one is how it is formed, is what kind of process, by modeling or remodeling. So this one we relies on animal studies. So this is mouse, and then we taken the left leg of the mouse, right leg of mouse subjected to daily mechanical loading for 5 days and 5 weeks. And the left leg serves as long loaded control. So these called rodent tibia loading models. And then what we decide to do is we do a weekly scanning of loaded and also long loaded limbs. So therefore, we started from -1 week, 0 week, and the first week that's where we begin to loading. The second week is 3 week, 4 week, and 5 week. And then we do another actual scanning in 6 weeks. So we have done the study. Weekly scanning do not cause significant bone loss. I mean, you can almost exact same. And then we quantify trabecular bone as well as cortical bone in the one scan. But I'm going to only reporting results on the trabecular for the time one. But with different scans, we do so-called registration, right? So therefore, you rotate all the images into a global coordinate system, register them. You can see all the images are rotated once here. And then you can subtract between the weeks. And then, you can subtract between the weeks, the images. So therefore, if we have, in week 2, you have less bone showing red, that's loss of bone. And if in week 2, you have more bone on the surface in green, that's bone formation. So therefore, in this process, we can quantify resorption and formation. But if we have 3 weeks moving forward, then not only you can quantify resorption and formation, you can also quantify modeling versus remodeling, right? So that's what it is. Because if you see there's background, the background suddenly have formation, that's anabolic modeling, just purely bone formation. And if we see resorption become background, that's catabolic modeling because the resorption did not follow bone formation. And if we see the resorption stop as background 1 week and the bone formation afterwards, that's a remodeling. So through this repeated scanning, we're able to do that. And this is showing example in the trabecular bone. A lot of activities going on. Is green is bone formation, red is bone resorption. You can see one the trabeculi on the top is called anabolic modeling. You can see the surface of the quiescent. Suddenly you have new bone formations in there. And in the bottom there, you can see there is a quiescent, there is resorption going on. The bone become a little bit less, and the following week actually add more new bones in there. So it's remodeling. And then the catabolic one is at the bottom left. And you can see the one trabecula actually resorbed, complete resorb, it's gone, right? So through this process, we can do that. And then you can quantify loaded versus unloaded, and they can quantify where it happens, what is in place, what is in rod. And then turn out, so this is a antibody modeling. You can see most of a bone response. This is a value is loaded, subtracted, unloaded of the same animal, and then you look at all the animal together. You can see the significant bone formation by anabolic modeling in the plates, but not in the rods, right? So therefore, mechanical loading actually creating bone formation, anabolic bone formation in the rod, in the plates, not in the rod. And then where the form, the plates, right? The plates is also mostly in the axial directions, can also be significant oblique direction and the transverse direction, but it's more in the axial direction. As I mentioned, axial direction is where subjected most mechanical loading. And then rod do not see too much changes, okay? Very few changes in the rod on the right side until you see the transverse direction. So indeed, they also have some transverse rod have active anabolic bone formations in here. We can also looking at remodeling. And the remodeling, indeed, remodeling is you have resorption first and the bone formation. Indeed, you also can create a significant adding more bones by mechanical loading through remodeling. And indeed, most of them is same thing, mostly on the plates. And the plates is also most of them is in the red in here, pink in here is in the long axial directions, some in the transverse directions, some in the oblique direction. So most of them is in the axial direction. Right? And then catabolic one seems like a catabolic resorption seems not depend on the mechanical. That's consistent also with the hypothesis resorption mostly is catastic, it's not a related mechanical loading, right? And then we'll really analyze how this data with the mechanical loading, right? We'll have all the scans, we actually can create a finite model for this one. So at least in the suggestion, we know in the compressive side, most of them is in the axial direction in both plates and rod. So axial direction, so therefore seems like an anabolic bone formation really targeting the axial loaded plate. And then in the rod, we know is most of the rod, the tensile side is on the transverse direction. So that probably also coincides with some of the transverse rod have active bone formation in there, especially in the anabolic ones here. So therefore, this is really what we're exploring right now is how to link in the detailed mechanical response in the tissue related ones here. So come together, therefore, increase anabolic modeling, remodeling, targeting plate-like trabeculi, and also transverse trabeculi. So anabolic actually turns about is majority response. You know, it accounts for 3/4 of bone formation by the mechanical loading. Okay, and then the 1/4 is from the remodeling process. And combine this together, I think the plate structure as well as mechanical loading, you're adding more bones in the plate, are designed similar thing as evolutions in here. Because as we coming out of the water, and we're still trying to loosen bones. And then if we're going back to the water, we actually maybe adding more bones. And then that one is actually in the evolution actually occurs. You can see the bone of fish is very, very solid until they get in the frog, that's amphibious. You can see lots of bones. Mouse is a mammal. You can see, bone become hollow, right? Pigeons, of course, you fly, your bone is even thinner. And then dolphins are mammals, but in the ocean. Why? They are land animal, mammals. And somehow, I think it's 10 million years ago, whale and dolphin decide they want to go back. When they go back and see, their bone actually become denser. So therefore, there's a reverse evolution here. So with the research we're doing in here, we found out, these two mechanobiology, bone microstructure, I think is related to the same questions in here. And then I'm going to be ending here, and thanks to my former student as well as many collaborators getting this work done, and happy to answer questions you may have. Okay, let me unshare such that we can see each other better. Thanks very much.
Thank you very much. That was a very, very wonderful talk. Do we have any?
Hey, Alan, good to see you.
Hey, Ed, how are you?
Good, good.
I do have a question.
Sure.
If you don't mind. So actually I have a bunch, but I'll just limit it to one. So the plate formation and the lateral struts forming in osteoarthritis.
Yeah.
Suggests to me that the local bone is trying to resist buckling. Does that make sense?
That could be. Yeah, that could be. Yeah.
And maybe because the cartilage thinning and the increased contact stresses.
Yeah.
And I just was thinking about that your cyclic loading model of the rat, the mouse, was similar to that in terms of results. How similar was that to an OA result?
Actually it is very similar because the loading apply is pretty high, in terms of getting response from the bone, right? So they could actually creating OA. So, but after we studied for a while, we actually are reluctant using mouse as a model for studying OA, especially focus on bones, because you only have like a 10 trabeculi in the subconjugal bone in the mouse. So therefore, what we would discuss is actually does not apply to the mouse, because you have variation, just change of one trabeculi, the results totally change. Yeah.
Right. That's cool stuff. And I guess getting down to the mechanobiology, and I was thinking about osteoporosis as well, where you seem to have just increased osteoclast activity and less osteoblast activity. And how all that goes together is pretty, it's very interesting.
Yeah, thank you. So I think, I think there's a coming together. Yeah. So we are doing, we did you can see we spent a lot of time doing OA for about 5 years now. And then you're in the biomechanics, even I'm doing bone, but I'm moving to OA. People say, oh, that's not your field. So I wouldn't get it funded yet. So we did as I say, we're not looking for mouse because they don't have too many trabeculi. So we need to look at the dog. But turns out the dog is actually not a good model either, because what happens, the dog, unless you're using very old dog, that's very, very expensive. Nobody's going to sell you the old dog. It's because the young dog, the epiphysis is not closed. So therefore, when you do not close the growth plate, the subconjugal bone biology is totally different. It's totally different. So now we're actually using a model, is guinea pig. Turns out, I showed a little bit of, the data on guinea pig. So we're using guinea pig now, and really for the guinea pig, we can do the exact same thing as we do in mouse. We can do weekly scans and follow up in the development of how they actually develop this OA and how the bone and the cartilage has been changed. So that's one of the graduate students working on right now.
Very nice. Well, very nice talk. I enjoyed it.
Yeah. Thanks, Alan. So I hope we'll see you in person next year.
Yeah, hopefully.
Yeah.
Do we have any other questions from our remaining audience members? Jay, I think you guys are meeting actually later, so. I wanted to ask a question about the differentiating between subpopulations of humans based on bone
Yeah.
scans.
Yeah.
Have you thought about looking into something like, particularly for the American, the Caucasian population, differences in either activity levels during adolescence? People who do sports maybe have better bone mass retention compared to those of us who lead very sedentary childhoods.
Yeah. Oh, absolutely, absolutely. I think it's I don't think we have data in the human subject, but I do know we have other people's data, not by our technique, but suggesting is plates probably is formed during this plates and rod is formed during the process of adolescent growth. Because you're starting with because the difference between the plates and rod, it actually occurs between the Caucasians and the Chinese girls actually occurs during the adolescent period of time. So therefore, the exercise of during the adolescent age, growth age actually is definitely critical. It's definitely critical. Yeah.
Well, thank you again. One last call for questions.
So that's always the challenge for the student, especially during this COVID Zoom, right? So one thing we do at our department when we used to have a seminar in the person, when I go there, I always challenge students. I say, we pay them lunch after the seminar. So I say, if no student asking a question, I'm going to take the lunch away, just joking with them. But that seems like stimulated them a little bit. Yeah.
That's how Vanderbilt actually used to motivate us too, if there would be lunch at seminars. So
that's right.
Well, thank you again, Ed. If you don't mind staying on for just a few minutes, and
yeah, sure.
Carrie and I can go over some paperwork stuff with you, and then Alan, thanks for logging in.
Yeah.
All right.
Ed, see you, Alan. Yeah.
Great to see you.
Good to see you. Good to see you. Absolutely.
I hope to see you soon.
Yeah. Keep safe. Yeah, safe.
All right. Bye-bye.
Bye-bye.